everybody. In this video, I'm going to be talking to you about gas exchange at the lung. We're going to be looking at lung structure, ventilation, and measuring lung volume. Now, the mammalian gas exchange system is something that you've covered at Key Stage 3 and Key Stage 4, so none of this is going to be particularly new to you, but we might go into a little bit more detail in certain areas. It's important to pay close attention at those points. So let's just have a look at the lungs in this diagram. So we can see we've got the nasal cavity and we've got the soft palate there at the top. And then we've got the trachea with its C-shaped rings of cartilage. Then the trachea splits up into two bronchi, the singular for each is the bronchus. Then the bronchi split into bronchioles, smaller vessels, and you can see how they still have these C-shaped rings of cartilage even quite deep down into the lung structure. And then the bronchioles end at these air sacs, these alveoli. So we can see the alveoli expanded here in a lot more detail, and we can see how we bring deoxygenated blood in from the heart, and we know that the pulmonary artery takes deoxygenated blood to the lungs. And we also know that the pulmonary artery is the only artery in your body that carries deoxygenated blood. And then the oxygenated blood goes back to the heart via the pulmonary vein. And we also know, if we're just trying to remind ourselves of the structure of the heart, we need to think, well, what chamber does the pulmonary artery leave? Well, the pulmonary artery leaves the right ventricle, and the pulmonary vein re-enters via the left atrium. So it's good to just have that context in your head so you can think about where blood's coming from and where blood is going back to. And then we can see how the alveoli are covered in this network of capillaries, this dense network of capillaries. And this is really important. Firstly, the deoxygenated blood is helping to maintain the steep concentration gradient between the higher oxygen concentration in the air in the alveoli and the lower oxygen concentration in the bloodstream. And the dense network of capillaries surrounding the alveoli is reducing the diffusion distance because the alveoli walls are 1 cell thick and the capillary walls are 1 cell thick. So that creates a short diffusion distance of travel for the oxygen to be brought into the bloodstream and the carbon dioxide to leave the bloodstream. So the other thing that the alveoli provide is an extremely large surface area. So that's the other factor that we know that any complex exchange surface needs is a large surface area to volume ratio, and the many millions of alveoli do that. The other thing that humans do to increase the rate of gas exchange is ventilation. We'll focus a little bit more on ventilation later on, but essentially we breathe in and out. We take in high oxygen content air and we exhale the low oxygen content air. And by keeping that movement of air into and out of our lungs, we are helping to maintain the steep concentration gradient. So that's the basic structure of the lung. Let's look in detail now at each part. So let's start with the trachea. So at the top of the trachea here, we can see the larynx, our voice box, and then we go down into the trachea. So the trachea has C-shaped rings of cartilage, incomplete rings, and you can see that here on this lovely microscope slide. You can see this ring of cartilage here. You can see it's not present at the back, and you can see the esophagus is here at the back. And this is the reason that the cartilage is not complete, is because if the cartilage were complete, the muscles, the smooth muscles in your esophagus would not be able to contract and relax to allow food to move readily down into your stomach. So we need to have these incomplete rings of cartilage. So what is the purpose of the cartilage? The cartilage keeps the airway open. Even if you have extreme changes in pressure, and that's exactly what happens. So say you take a really big breath in all of a sudden, or a really big breath out all of a sudden. This would be a rapid change in pressure inside the trachea. Without the cartilage, the trachea would collapse in on itself. And because it's moist, the walls would stick together and you would be unable to reopen them and you would suffocate and you would die. So the cartilage keeps your air passage open even during pressure changes. So they're really important. So the cartilage rings are separated. So here's our cartilage ring, and then they are separated by muscle and elastic tissue. This muscle allows contraction and relaxation of the trachea to increase and decrease the volume of the air passage. And the soft tissue, so the elastic tissue, allows the trachea to be able to stretch and recoil in turn changing the size of the airspace of the trachea to allow more or less air through. So let's talk about the bronchus. The bronchus is the first branch of the trachea, and it allows the passage of the air into the bronchioles. They still have cartilage in the walls to keep them open. So if you take a look, you can see here that there's cartilage in the walls, but it's not this C-shaped rings of cartilage, it's just cartilage is present. The other things that you see in the bronchi is that you've got ciliated lining of the bronchus. So if we were to look at cells lining the bronchi, we would see that they have cilia, they have fine hairs that move in a mechanical wafting motion. So you also have cells that make mucus. So can you see these gland ducts? So we have these mucus producing cells in the bronchioles and in the trachea. And so the mucus lines the cells, and then any dirt or dust or bacteria gets trapped, and then the cilia waft the dirt and the dust and the mucus up towards the back of your throat, and then you swallow it down in your stomach. This is there to protect you from getting any infections in your lungs, and it goes down into the stomach where the hydrochloric acid will kill any potential pathogens. So the bronchioles are the smaller branches of the bronchi, and they end in these clusters of alveoli, like these bunches of grape clusters of the alveoli. Now, the smaller bronchioles have only muscles and elastic tissue, but the larger bronchioles still have a little bit of cartilage in them to keep them open. So the muscle and elastic tissue allow them to contract and relax. Just in terms of terminology here, muscles allow them to contract and relax, and elastic fibers help them stretch and recoil. And this is really important during ventilation, so during the mechanical movement of air into and out of your lungs. So let's talk about gas exchange in the alveoli. So oxygen from the air moves down the trachea, bronchi and bronchioles into the alveoli. This movement happens down a pressure gradient. So air moves into your body when there is a higher pressure in the atmosphere in comparison to in your thorax. So air moves down its pressure gradient into your lungs. Once in the alveoli, the oxygen diffuses across the alveolar epithelium by simple diffusion. So gases can move by simple diffusion, and then across the capillary endothelium. Each of these linings is one cell thick, ending up in the lumen of the capillary itself. This is happening down a diffusion gradient. So there's a higher concentration of oxygen in the alveolar airspace, and a lower concentration in the capillary, and so it moves in. Once it gets into the capillary, most of that oxygen is picked up by red blood cells and forms oxyhemoglobin. So the alveoli are adapted to speed up the rate of diffusion. We talked already about the fact that they're a single layer of cells. So a single layer of cells on the alveoli, a single layer of cells on the endothelium of the capillaries for a shorter diffusion pathway. They have a really large surface area, so millions of alveoli allow gas exchange to occur. And they have a really extensive network of blood capillaries, and this maintains a steep, large is not a great word here, I'm just going to change this to steep. This maintains a steep concentration gradient. So the walls of the alveoli are fully permeable to gases, so the gases can just move directly across by simple diffusion. Now, the inner lining of the surface of the alveoli is moist, it's lined with a layer of water. Now gases diffuse slower in liquids than they do in gases diffuse slower in liquids than they do in air. So why would we have this watery layer? Well, the oxygen is unable to enter the cells directly from the air. Gases cannot enter a membrane directly unless they're already dissolved in a solvent, i.e. water. So the water is there to allow the gases to dissolve in that water to allow it to move across the membrane. In addition, there are cells lining your alveoli that secrete a solution called surfactant. Surfactant is a solution which lines your lungs, and lowers the surface tension of the water. So you need the water to allow the gas to dissolve to be able to travel across the membrane. But the problem with water is water has high surface tension. Water molecules like to stick together. So when there is a pressure change in your lung and the alveoli might all the alveolar walls might all come closer together, that surface tension in the water might cause that water to stick together. And if the pressure inside your lung can't break that surface tension, your alveoli are basically constricted. You can't expand them back out. You can't do gas exchange, and you're going to suffocate and die. So surface tension is a danger on the inside of your lungs. You've got to have the water there, but the problem with it is high surface tension. So your lungs secrete this substance called surfactant that lowers the surface tension of the water. This means that your alveoli will not stick together during pressure changes. So surfactant is really important in your lungs to enable your alveoli to remain in shape. So I'll just give you a minute to read through this information, which summarises what we just said. So I thought it would be good just to have a look at this microscope image of the lungs. So we can see here the internal surface of the alveoli. So we can see all these alveolar membranes here. And we can also see the bronchioles. So we can see the bronchioles here. We can see the ciliated linings. And we can see some blood vessels with the blood inside. So let's talk about ventilation for a minute. Ventilation is the movement of air in and out of your lungs through inhalation and exhalation. So we've got inspiration and expiration. So ventilation is controlled by the movement of the diaphragm, this sheet of muscle at the base of your lungs, your internal and external intercostal muscles. These are the muscles that sit in between your ribs on your rib cage. So if you were to go and eat some ribs, you would actually be eating intercostal muscles. And the movement of the rib cage itself. So ventilation in humans is tidal. What that means is the air moves in and out through the same space. So ventilation is tidal. So air moves in and out of your body in the same way. So in fish, it's not tidal because the water moves in through the mouth and out through the operculum. But in insects, it's tidal, in and out through the spiracles. And in humans, it's tidal, in and out through the trachea. So let's just take a look at this diagram to see that we understand the terms internal and external intercostal muscles. So your internal intercostal muscles are the muscles on the inside, the ones closest to your lungs, the innermost intercostals, like they're called here on this diagram, the internal intercostal muscles. So that's these here. And then you've got the external ones, the ones that are on the outside, so internal and external intercostal muscles. Try and visualizing that in your head so you understand what that's talking about. These are an antagonistic pair of muscles. Most of the muscles in your body act as antagonistic pairs. What that means is as one contracts, the other relaxes, and vice versa. So let's think about the process of ventilation. Breathing in is an active process. That means not just that it uses energy, but that it uses energy from respiration. So it uses energy in the form of ATP. So when we breathe in, the external intercostal muscles contract. And its antagonistic pair, the internal intercostal muscles, relax. This contraction of the external intercostal muscles pulls the ribs up and out. This increases the volume of the thorax. At exactly the same time, the diaphragm muscles contract. This causes the diaphragm to flatten and also increases the volume of the thorax. The increased volume of the thorax results in a reduction of pressure. This is Boyle's law. An increase in volume will decrease the pressure, and a decrease in volume will increase the pressure. Now, what you've done there is you have created a pressure gradient. So now the atmospheric pressure is greater than pulmonary pressure, and air is forced into the lungs. We draw air into our lungs down the pressure gradient. Now let's think about expiration. Breathing out is a largely passive process. That's kind of an odd statement, largely passive. Well, it kind of means two things. One, when you just sat still, you're not really doing anything. Most breathing out just relies on the recoil of the elastic tissues in your lungs. So you don't really put much effort into breathing out. It's only when you are doing something strenuous, so when you are breathing out forcefully, that you really need to engage your diaphragm and your internal intercostal muscles. So mostly it's passive, sometimes it is active, and it is active when you are doing something strenuous. So let's just talk through expiration. The internal intercostal muscles contract, and their antagonistic pair, the external intercostal muscles, relax. This causes the ribs to move downwards and inwards. This decreases the volume of the thorax. The diaphragm muscles relax and it goes up, so it forms a dome shape. It gets pushed up by the contents of the abdomen, and the volume of the thorax is further decreased. The decreased volume of the thorax increases the pressure. So now you've changed the pressure gradient. Now the pulmonary pressure is greater than that of the atmosphere and air is forced out of the lungs. So one thing you might want to measure is your pulmonary ventilation rate. The pulmonary ventilation is the volume of air taken in per minute. The way that you can determine your pulmonary ventilation is by first calculating your tidal volume, which is the volume of air normally taken in with each breath, times by the ventilation rate, i.e. the number of breaths taken in 1 minute. So you can obviously just count the number of breaths taken in 1 minute. But how do we calculate the tidal volume? In order to calculate the tidal volume, we use a machine called a spirometer. So let's take a minute to have a look at that now. So this rather odd contraction is a spirometer. So the general principle behind using a spirometer is really simple. It's effectively just a tank filled with water in an air-filled chamber suspended in the water. It is set up so that adding air to the chamber makes the lid of the chamber rise up, and removing air makes it fall. So as this person breathes out, he adds air to the chamber. Adding air to the chamber will cause this floating drum to rise. When the floating drum rises, this is attached to a little piece of string all the way down here. And so the lid rises, the string goes down, and the line goes down. And then when he breathes in, he removes air from the chamber. This causes the lid to come down. That pulls the string with it. And now this little line is going to go up again. So this line will correspond with his breathing in and out. And this drum is continuously rotating so that the line will go along. This will produce something that looks a bit like this. So this is a spirograph. This is showing the pen moving along the paper as the person breathes in and out. So the first thing we have here is your tidal volume. Your tidal volume is the normal amount of air breathed in and out during a normal breath. Then we've got the vital capacity. So in this situation, you might be asked to take a really deep breath in. And when you take a really deep breath in, you inhale a lot more air, and a really big deep breath out. Now, you can never breathe out all of the air in your lungs. This is because your lungs have residual volume. Residual volume is just the air that you just don't move in and out of your lungs. So this data could help you calculate your pulmonary ventilation. This is sometimes referred to as minute ventilation. So minute ventilation equals tidal volume times rate of breathing. Okay, so I hope that's been helpful. Well done for listening, and I'll speak to you soon.